So while you soak in this little life hack, I'll remind you that in our last screencast, we looked at DNA as a molecule. We looked at its structure. We looked at how it replicates. In this screencast, we are going to take a look at something called protein synthesis, which is really all about how does DNA actually do what it does. When we say DNA codes for something, how does it actually code for that? How does it result in brown eyes or blue eyes or purple flowers or whatever it is? That's what this screencast is all about. So before we can really get at that, it's important to review some key concepts that we learned earlier in the year. So I'd like you to take a moment and reflect on the following questions. What do proteins do in the cells? What cell structure builds proteins and where are those cell structures located? And then what are the building blocks of proteins? How do they arrange themselves to form a protein? So hit pause, take a moment to reflect, and we'll discuss. So what do proteins do in the cells? List 2 to 3 functions. So the answer is they do a lot. Big ones that we've discussed is they act as enzymes. And we saw, that basically every step of glycolysis and the Krebs cycle was carried out by an enzyme. The electron transport chain is basically this collection of proteins in the membrane of the mitochondria that help carry out reactions. They allow substances to enter and exit cells. So we saw that just basically facilitated diffusion or active transport. And then also, again, in that electron transport chain, whether it's in the mitochondria or chloroplast, we saw that some of these proteins are proton pumps that allow protons to pass through the membrane through there. We saw that proteins are involved in moving things around. So vesicles, you may recall, are materials that transport things throughout the cell, and there's these little motor proteins that kind of move the vesicles along the microtubule tracks. So when we looked at how proteins go from the endoplasmic reticulum to the Golgi apparatus, we said that they pass between those things through the vesicles, and the vesicles are transported by motor proteins along these microtubule tracts, which are part of the cytoskeleton. The spindle in mitosis and meiosis, those are proteins. We found that there's structural components of cells. So again, the cytoskeleton that kind of gives support and shape to the cell, that is protein. And then we saw all sorts of other things. We saw a role for them as receptors in cell signaling, and all sorts of other things. So proteins do a lot. That's the bottom line. They do a lot. What cell structure builds proteins and where are those located? Well, hopefully you remember, they are built in the ribosomes. Now, we haven't talked much about ribosomes, but we did mention 2 places where they're located. One is on the rough endoplasmic reticulum. These little green dots represent ribosomes. And the other place, not really indicated on this figure, but they also float freely in the cytoplasm. So if a protein is destined to just sit in the cytoplasm, say, for example, a enzyme involved in glycolysis, which takes place in the cytoplasm, then a free ribosome in the cytoplasm would produce it. If that protein is destined for, say, the lysosome, maybe it's a hydrolysis enzyme, then it's going to be produced in a ribosome in the rough ER, transported to the Golgi apparatus, and then delivered to the lysosome. What are the building blocks of proteins, and then how do these arrange themselves to form a protein? So the building blocks are amino acids, so-called because every amino acid consists of a central carbon with a hydrogen, and then an amino group and a carboxylic acid group, so amino acid, and then that R group. I want to remind you now that there are 20 different types of amino acids. All of them vary based on that R group. So shaded in blue here, you see the central carbon, the amino group, the carboxylic acid, and the hydrogen. And then in yellow is the unique R group that gives each of the 20 amino acids its distinct identity. And if you look closer at these, you'll see that some of these R groups are non-polar, some of them are polar, some of them have a charge, some of them are acidic, some of them are basic, and so there's different types of amino acids that are going to behave differently based on that R group. To build an actual protein, those amino acids are connected in a very specific order. So we join 2 amino acids together through a dehydration synthesis reaction, and we form what's called a peptide bond there between one amino acid and the next. And then those amino acids, once they're joined together, are going to form what is called the primary structure of a protein, this specific sequence of amino acids. Again, it's not random, it is a very specific sequence of amino acids that makes up the primary structure of that protein. And then because of those little R groups sticking out, not to mention the amino group and the carboxylic acid, the protein starts to fold in on itself based on the interactions between those chemical groups. And so we can see little hydrogen bonds forming between the amino group of one and the carboxylic acid group of another. And that creates these sort of localized structures, the alpha helices, the beta pleated sheets, and then ultimately it folds itself into this very specific three-dimensional shape. And since this is complicated and hard to draw, we draw it like this. We kind of show that very specific three-dimensional shape in this schematic way to help us understand how, say, an enzyme works or how a protein receptor works in cell signaling or whatever it is. So that is how the protein actually does its job. You may also recall that sometimes several of these peptides come together to form a larger structure, and that's the quaternary structure, if you remember that. So then the last little point I wanted to make is that there's 20 types of amino acids, but each of them is arranged in a very specific order to form a specific protein. And so by arranging the 20 types of amino acids in different combinations, you get a huge diversity of proteins, much like the letters of the alphabet. You arrange these 26 letters of the alphabet, and you get a dictionary worth of words. But this analogy falls short a little bit because this dictionary is only English words, and there's many different languages. And not to mention that most words are probably what, I don't know, 5 to 7 letters-ish long, whereas proteins can be hundreds, thousands, tens, hundreds of thousands of amino acids long. So the amount of diversity in proteins is truly staggering, absolutely mind-boggling. So with that background, we can now begin to understand the importance of protein synthesis, this idea of building proteins. So, quick overview. We know that the order of amino acids is important. That's what kind of determines the primary structure, which ultimately determines how it folds into a specific shape and what job it does. So the questions for this unit are, what specifies the order of amino acids? And then how does the cell put the amino acids in the correct order to actually assemble the proteins? Well, the answer to that first question is DNA. The DNA is what specifies the particular order of amino acids in a proteins. In other words, proteins are the link between genotype, what the DNA says, and phenotype, what's actually happening in the cell, what's being carried out by those specific proteins. That's a big deal. We should celebrate. Woot! So DNA, the importance of DNA is that it tells us what order to put these amino acids in. And once that order is determined, that affects how it folds up into that specific shape, what the active site is going to look like, and what that protein is going to do in the cell. Again, DNA is the link between genotype and phenotype. Woot. So how does it do that? Well, that's where things get a little bit more complicated, and that really is the focus of this unit. So let's pause for a moment, and let's go to our correspondent on the ground, Mr. Sanger, and draw out a little analogy, baking a pie. Thank you, Mr. Sanger. So behold, ladies and gentlemen, this circle represents the city of Edina. Now I want you to imagine that you are in the city of Edina, and you decide that you would like to make a cake for a dear friend's birthday. Now, the first thing you're going to need to do is figure out how to actually make a cake, because I'm guessing most of you don't know. That's too bad. Go learn how to make a cake. And so where are you going to go? And for this analogy to work, the internet is down. So basically, in order to make a cake, you are going to need a cookbook. Okay? And the place you're going to go to get a cookbook is the library. So I'm going to draw the library of Edina, fairly large, inside my city. Now, the cookbook looks like this. Kind of a weird looking book, right? We'll just kind of give that cookbook a nickname. We'll call it DNA. Okay. So DNA is like a cookbook. It has recipes to make pretty much anything you would ever want. So you go to the library, you grab the book, and you go to checkout and leave. And you go to the checkout desk and you say, "Hi, I'd like this book." And guess what happens? The librarian says, "Oh, I'm sorry, this book is not allowed to be checked out. This is for reserve only." And you're like, "What? Are you kidding me? I can't leave the library with this book?" And the librarian's like, "Sorry." So you make a break for it, you sprint, the little like alarm thing goes off, bam, 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 and then all of a sudden the security guard tackles you. There's no way out. You cannot get out of the library with this book. So what are you going to do? Well, what you're going to do is you're going to make a copy. You're going to say, all right, if I can't take this library book out, then maybe what I'll do is I'll just make a photocopy. Now, obviously, you don't need to photocopy the entire, 300-page book filled with recipes for everything on the planet. Instead, you're only going to photocopy that one little recipe for the cake that you're trying to make. And so you are going to take that DNA and you're going to make a little photocopy that looks like this. That photocopy we are going to call mRNA or messenger RNA. Again, it's much shorter than the cookbook because it's just the one recipe. Now, with that, you are actually able to leave the library and go to other parts of this grand city of Edina. And the specific place you want to go in this grand city of Edina is you're going to want to go to your kitchen because that is where you're going to make a pie. And so the kitchen, it kind of looks like that, and we're going to nickname that thing the ribosome. And so what happens is you take your recipe and you go to your ribosome, and I'm getting like too many markers going here. And in that ribosome, you are going to like feed in your photocopy of messenger RNA. In other words, you're going to take that recipe, and in that ribosome, you are going to use it to make a protein. I don't know what color to use, so I'll just call it this. Okay, so you're going to convert the messenger RNA to a protein. In other words, you're going to make your cake. So we start with the recipe book, the cookbook that has recipes for just about everything. We photocopy the one recipe we need. That one recipe is called the messenger RNA. Both of these are information. They're just telling us how to do things. And then we take that recipe to our kitchen, the ribosome, and use it to actually bake our cake, the protein. And the cake is what's actually going to do the thing in the grand city of Edina. That is, it is going to make you a happier friend and a more popular person because everyone loves cake. And even though you're just buying their affection through cake, you don't care. Okay, back to you, Mr. Sanger. Thank you, Mr. Sanger, on that wonderful life lesson on how to buy friends. So to recap, here's a little bit nicer diagram that just kind of shows this. So we start with DNA, the cookbook, and the library in that previous analogy represented the nucleus. The DNA stays in the nucleus. It's too big to pass through those nuclear pores, can't get out, so it just stays put. That's your cookbook, has recipe for everything that the cell needs. Then this process called transcription occurs that takes that DNA and converts it into something called messenger RNA. That's like a recipe. It's just a little disposable copy of some of the information of DNA. We then are able to leave the nucleus with that smaller recipe that can fit through the nuclear pores, where we go to the ribosome, the kitchen, and that's where we do this process called translation to actually make the protein. And again, this is the link between genotype, the information in DNA, and phenotype, the physical manifestation of what's actually happening in the cell. So in our next screencast, we're going to take a look at how this all takes place. We'll catch you then.